Mapping the genome. When we say the genome, we're talking about genomics. That's going to be the entire genome. So all the genes, the ones that are expressing, the genes that aren't really coding for anything that we might call junk DNA, all the nucleotides and their interaction is genomics. So we want to see what the genes are. Let's map them out on the chromosomes. So we're going to make maps. There's two maps. There are genetic maps, and genetic maps are going to have the genes and their location on the chromosome. Breast cancer is located here. And yeah, here's breast cancer. Alzheimer's is in this location, which is on the same chromosome as this trypsin deficiency, also on the same chromosome. So genetic map is going to tell you what the genes are and where they are on the chromosome. Genetic markers are going to look at this specific gene and how likely it is to be grouped together with another trait. So this gene and this gene, are they linked? Are they going to have a certain recombination frequency? So that's genetic markers. And so ways that we might look for genetic markers would be looking at rift flips or variable number tandem repeats, microsatellites, or single nucleotide polymorphism, SNPs. All of these are different things that will help us map out the genome. And so when we're talking about mapping it out, we said, you know, this chromosome is located, this gene is located on this chromosome here, like we've got in the Human Genome Project. The Human Genome Project is something that was a little bit of pure science and a little bit of applied science. We want to know what the genome is for people just to know, but we also can apply that knowledge towards maybe treating those genetic diseases. So it's pure and applied science. You can find out more about the project. It took 13 years to map out all of the genes in our chromosomes, but you can find out more about it at the website for Human Genome Project. So it's going to incorporate genetic map, which are the genes. The markers are this gene is associated with this gene and what's its recombination frequency. How likely is it that these things will be linked? Remember, we talked about recombination frequency before, where the closer the things are, the more likely that they will be linked and their recombination frequency will drop and it will be low, versus others where they're further apart on the chromosome like these guys are, then crossover is more likely to occur and recombination frequency will be higher. Recombination frequency is low if they are linked, so closer to zero if they are linked. So these guys, we have, you know, B and C are close together, so they may transfer as one big unit to the next one because they're linked. So what's their recombination frequency because they're linked? Those because they're linked are genetic markers. And then mapping those things, we have them in this picture here from the textbook, which mentions this study with fruit flies. And so I pulled it up off of Wikipedia and added this into the PowerPoint, where you can see that they're comparing the different genes and where they are on the chromosome, so we can see if they're linked or not. Are they close together and they're linked and their recombination frequency is low? Are they farther apart on the chromosome and the recombination frequency would be higher? So short or long antennas, black body or a gray body, red eyes or the cinnabar, normal wings or the kind of short, crinkly vestigial wings, red eyes and brown eyes. So you can see all of these different things with the wild long wings versus crinkly vestigial wings, different body colors, all sorts of things that can be mapped genetically. What about the physical map? So the textbook also mentions physical maps, and those are going to be the distance apart. This gene is on the chromosome here, this gene's on the chromosome here. How many nucleotides are going to separate this gene from this gene? That's a physical map. You can see the physical distance between the genes is physical map. So a type of physical map, there are three different ones. There are cytogenic, there's radiation, and sequencing. This is a cytogenic map right here. And we can see, you know, here we'll look at, I don't know, chromosome 13. And on chromosome 13, we can see that there are, based on what the way the DNA absorbed the dye, and it is reflecting back on this map, we can see that this one is a light color and this is a light color. So those are closer together, and we can see, measure the distance out on a cytogenic map based on their stain. On the radiation map, we're going to use radiation in the form of like X-rays to break DNA up, because we said X-rays can cause DNA mutations and changes to DNA. So if we use radiation, we can form a radiation map and break up the DNA into fragments and see how that works. Or we can use sequencing, which we're going to talk about in the next section, where we have figured out what all the nucleotides are, and now we can actually map out the distance based on that knowledge. So genetic map, physical map of these guys.